Hello, everyone, and thank you for joining us for today's webinar, Neurotransmitters and Communication Between Neurons. My name is Danny Shapiro, and I'm on the marketing team here at Hawks Learning. Our speaker today is Dr. Laurie Slifka of Keiser University. Dr. Slifka has authored questions for our course, Introduction to Psychology. We arranged this webinar so that she can share some of her expertise with you, other psychology instructors from across the country. At Hawks, we understand the value of partnering with faculty in order to create and maintain high-quality course materials. Excuse me. We believe that your expert insight is invaluable, and that's how we were able to connect with Dr. Slifka. Dr. Slifka is a research neuropsychologist with specific expertise in health conditions such as human immunodeficiency virus and early life stress that primarily impact brain structures deep beneath the surface of the cerebral cortex. A majority of her research studies incorporate neuropsychological tests and structural neuroimaging. She has also developed research protocols and statistical analysis plans, designed neuropsychological batteries, and created databases for integration of clinical, neuroimaging, and neuropsychological data. She has been funded by the National Institutes of Health for her work investigating brain integrity in individuals with human immunodeficiency virus. Additionally, Dr. Slifka has contributed to numerous peer-reviewed publications and presented work at international conferences. She currently serves as an ad hoc reviewer for several peer-reviewed journals, including Psychiatry Research Neuroimaging, Behavioral Brain Research, Journal of the International Neuropsychological Society, and Neurobiology of Aging. Dr. Slifka has a passion for teaching and utilizes her abundant training and research experiences to inform and greatly enrich her teaching and mentoring practices. We're so excited to have you, Dr. Slifka, for the third webinar in the series that you have done. Audience, if you have questions as we go through the presentation, please enter those into the Q&A box, and we'll talk about them at the end of the presentation. So on that note, I'd like to hand it over to you.
Thank you so much, Danny, and thank you, everybody, for coming. Today, I'm going to be talking about neurotransmitters and communication between neurons. For those of you that were able to join for the last webinar, this is an extension of that. So if you weren't able to make it, I recommend that you go into Hawks and you check out the prior recording where we talked about communication within neurons. So we're essentially taking it a step further today. Danny provided such a wonderful background, so I don't want to spend too much time on this. So just a little bit of additional background. I got my PhD in psychology with an emphasis in behavioral neuroscience. I'm currently a professor of psychology at Keiser University. I actually do teach online. I teach grad students, both master's and PhD in the psychology and industrial organizational psychology department. I know a lot of you are transitioning right now from on-ground to online. I know for some of you it could be a little bit frustrating. I know I had a big learning curve when I started, but just some words of encouragement that have nothing to do with the brain. Just hang in there. We're all in this together. We got this. If you do have any questions about online teaching, please feel free to ask, to send me an email that I'll provide you at the end. I'm happy to help in any way that I can with that, because I know it can be a difficult challenge. Here's numerous topics that I've studied throughout the years. Danny did a nice introduction of that, so I don't want to go too deep into that. But essentially the way that I'm able to study these conditions is through two main methodologies. One is neuropsychological testing, so essentially doing cognitive testing to test people's thinking, and then neuroimaging with a specific focus on the structure of the brain. So we have both structural neuroimaging and functional neuroimaging. So I focus on what the brain actually looks like, and specifically what the pathways that go from one area of the brain to another, how they're doing. So I don't look at those big macro structures. I look at the roadmap or the connections going from one area to another, which is very fitting because this is actually the connection of neurons, which we're going to be talking about today. You all may be a little bit more interested in my research than my students. I know some of my students are, some aren't, but I always tell my students, if you need a good bedtime story, here's my research profile. So, feel free to check it out if you're suffering from any sort of insomnia. I always like to provide an overview so you guys know what's to come, so you know when you want to get up and skip any parts or whatnot. I'm kidding, it's hard to try to make jokes when you can't see people. You don't know if the audience is really there, but I try. So we're going to do a bit of a review of communication within neurons. So we're going to go over what was discussed in the last webinar for those of you that were not able to make it. For those of you that were, it's a little bit of a refresher. It never hurts to hear again. Then we're going to talk about communication between neurons. So that's the focus of today, what happens from neuron to neuron. And then we're going to talk a little bit about some neurotransmitters and specific receptor types. I'm not going to go too deep into all the different neurotransmitter types because it can get really, really complex. But if you're teaching an intro to psychology course, possibly an intro to behavioral neuroscience course at the undergrad level, this could be, you know, something that you would do with your students, kind of like an intro review to neurotransmitters without getting too deep into the individual mechanisms. So like I said, we're going to start by reviewing communication within neurons. So if I were in a typical classroom right now, I would ask, what do I mean when I say that? What do I mean when I say communication within neurons? Looking for a specific word. For those of you that are shouting it at home, your husbands and wives and dogs are like, what are you saying? Action potentials. So essentially communication within neurons is, we're talking about action potentials. Okay, so what we did last time is we went over the structure of a typical neuron. So this is what a typical neuron looks like. Not every single neuron looks like this, but if you Google neuron, this is probably what you're gonna get. I always describe the neuron in terms of a hand model. So I don't know if you can see my hand. I turn off my webinar so I can't see myself talking. But the dendrites are my little fingers here. The cell body, the soma is my palm, and then the axon's my arm. So I kind of think of it like this, okay? I don't know if that helps or not, but so the main parts, just a very brief overview. The soma or cell body is the factory of the neuron. So it's where all of the important parts are to keep that cell going. So the nucleus is extremely important. It's in the middle of it. It contains our genetic material, including information for cell development, synthesis of proteins necessary for cell maintenance and survival. The mitochondria is the energy provider for the cell. It's surrounded by a cell membrane or neuronal membrane, which we're going to come back to in a bit, but it serves as a barrier to enclose the fluid within the neuron from the fluid outside of the neuron, so it's keeping everything in there. The dendrites, so if we're going outside the cell body, our fingers are the dendrites. I like to think of this as the listening end, so it's hearing and receiving signals from other neurons. So it's the main apparatus for that, for receiving the signals, okay? Then we get into our arm. So the axon, which runs down here, I like to think of it as the talking end, because it's typically what's sending signals to other neurons. And it's covered by what's called myelin sheath. Many neurons are. This helps speed up that transmission within an individual neuron. Those nodes of Ranvier that are in the middle help with that too. It can help the connection kind of jump from node to node, which speeds that up, which is good for fast impulses throughout the brain. And the axon terminal, which refers to the end of that axon, it is where our communication point starts between one neuron and another. Okay, so the communication. So essentially, we start at the dendrites, our little fingers, that's the listening end, and then information is sent through the neuron out the talking end, okay? So there are multiple different classes of neurons, but this is what we think of as our traditional neuron. Receives signals, it integrates incoming signals, so it may be getting multiple signals at the same time, and it communicates the signals to other cells. In a very elementary explanation of this, an action potential, which is what the communication is, occurs when a neuron sends information down the axon away from the cell body. We may use the word spike or impulse for an action potential. It's an explosion of electrical activity that's created by what's called a depolarizing current. So a cell becoming more positive. We're going to talk a little bit more about that. So the way that a cell is at rest. The cell is maintained based on the concentration of ions, both inside and outside the cell. So at rest, the cell is typically negative, has a negative concentration based on the distribution, sodium and chloride and potassium. So potassium are typically found in higher concentrations within the actual cell. Sodium and chloride are typically found in high concentrations outside of the cell. This balance of it creates this negative charge. So typically when a neuron's at rest, it's not firing, it's around -50 to negative 75 millivolts. That will come into play in just a little bit. And this axonal distribution is maintained, or this ionic distribution is maintained by something called the sodium-potassium pump. It's what's helping that distribution stay as it is to keep the cell negative, and it helps return the cell back to that resting position after an action potential fires, okay? So you've seen this before. This is in every introduction to psych textbooks. When I first looked at this, I was like, what? Like when I was taking intro to psych, I'm like, what? This is way too complex for me. This is intro to psychology. I don't want to be learning about this stuff. It took me a little bit to understand it. And it may take students a little bit to understand this information because it can get complex. And you know, some of them dread this chapter. They dread the neuroscience one. But I think if you really are able to break it down for the students, really explain it, give them resources to go over it, eventually they will be loving neuroscience. So essentially an action potential occurs when a neuron sends information down the axon away from the cell body. Okay, so this is what's happening during that process. This is what we're seeing at like the micro level, essentially. So an action potential is an explosion of electrical activity created by what's called a depolarizing current. This means that some event, so some stimulant or stimulus that acted on this causes the resting potential to move towards the -0 millivolts. So usually we would think it going to negative that it would be, or 0, it would be getting negative. But in this case, we know it starts negative based on the distribution of ions. So it's becoming more positive. This is a depolarizing action. You can see that slope go up, okay? So once it reaches, so there has to be a certain amount of stimulus for it to reach what's called a threshold of excitation. We can see that dotted line around -55 millivolts. If it hits there, an action potential will fire, okay? It's always a fixed fire. There's no like really big action potential or really small action potential. An action potential takes place if it reaches that threshold of excitation, okay? So the way that this happens is this whole process occurs because of different ions crossing that neuronal membrane. So that barrier, essentially. So a stimulus will first cause the sodium or Na, or you see Na positive, to open, okay? Because there's so many more sodium ions on the outside. The inside is negative to the rest of the outside. The sodium's rushing to make it more positive. They're positive, so they rush in. They're trying to make that cell more positive, okay? So this is that depolarization action. And then it takes a little bit longer, but then the potassium channels open. When they do, potassium rushes out of the cell, reversing that depolarization. So it's automatically bringing it back to rest. It wants to go back to that point. This is the firing, okay? About the same time, the sodium channels will close. So you're already starting that returning to baseline charge. And then this causes the action potential to go back to -70 millivolts. And then at one brief time, it goes beyond -70. This is called a hyperpolarization. So becoming more negative. And this is because the potassium channels stay open a little bit too long. But then gradually the ion concentration goes back to rest at our -70 millivolt level. So this action that takes place starts the process of the communication that happens between the neurons, which is what we're going to get into now. So you guys seen something that looks like this? I always think, so this is an image of diffusion tensor imaging, which is the neuroimaging technology that I use, but still, I have done my own research. I have created my own images. I still think of the cover of Muse I don't know if you listened to the band Muse, but they used a diffusion tensor imaging scan. So this very pretty picture, it's essentially just telling us directionality of the flow throughout the brain. But there's all these individual neurons and they're communicating with each other. This communication that takes place is what's leading to this. This is leading to communication that leads from one area of the brain to the other. It's essentially what I like to think of as our highway system. So in our brain, we have these large structures, these large gray matter structures. So if you've heard hippocampus, the amygdala, the hypothalamus, frontal lobe, it's all these like gross structures we think of. This communication that's taking place between the neurons is what's sending signals between those areas to coordinate all this activity that's taking place in our brain. So much activity taking place at one time. Think about everything that I'm doing right now. You may be like, you're really not doing much. You're just sharing the slides with us, but I'm communicating with you guys. So I'm talking, I'm bringing up information that I've learned in the past. I'm seeing, I'm hearing, I'm using my hands. So all this information is in, and I'm breathing. I'm breathing, my heart's beating. And so all this is happening simultaneously because of this communication that happens throughout the course of the brain. So hopefully I got you extremely excited about it because that's what we're gonna get into now. All right, so communication between neurons, also known as synaptic transmission, okay? So now we're taking what, we think of two neurons and we're marrying them, or they're kissing, essentially. I don't know if I've ever heard anybody describe it like that, but that's kind of what's happening. They're communicating together. We're going to talk about it like we're looking at typical neurons. For a review on the different types of neurons, I highly recommend that you watch the prior recordings. I don't want to get into that, but I do want to just talk about it on like more of a basic level. So if we're going from axon sending information to dendrites, okay? That's where my head is. So just telling you that so you get on board with that. That's the way that I'm thinking about this, although it's not always the case. So in most cases, these action potentials we just discussed are not transmitted from one neuron to another, okay? That goes back to our title. It's communication within neurons. So how is information sent from one neuron to another to create that beautiful highway system? Well, the information is communicated at what we call synapse, the synapse, the space in between the axon of one neuron and the dendrite of another neuron. So information is sent from what's known as the presynaptic neuron, which pre-signal, so the one sending the signal, to the postsynaptic signal through the synapse. We're going to go into more definitions of this. But what we see in most cases, and again, I'm talking about this more generally, there are individual cases, there are different types of neurons, there are different types of neurotransmitters, but just at the most basic level, a neurotransmitter sends this chemical signal from the presynaptic cell to the postsynaptic cell, okay? So if we were in a traditional classroom, I know many of you are getting used to this. I'm still kind of getting used to all of this too, so these pauses are where I would usually be like, does anybody have any questions? I know in a platform, in a classroom, you can do that. But so if you hear me pausing, it's because I'm like, okay, let's touch base here. But we're all professors, so it's a little bit different. I still have that natural instinct. All right, so a synapse, okay? So if you look at the picture on the right, synapse is essentially that space. It's a place where information is transmitted from one neuron to another, okay? There can be chemical synapses, which is what we mostly think of when we think of communication. That means that a neurotransmitter is being transmitted from one to another. And electrical, not as common, but very important for survival. We will talk about that as well, okay? So a presynaptic neuron, like I said, is a neuron transmitting the signal. So I don't want to test my luck here with this technology. I'm still getting used to Zoom, so I'm not going to. So I'm just going to walk you through it. But if you look at the image on the right, that top portion looks kind of like a monster that's going to eat the bottom portion. That top portion is our presynaptic cell. It's the end of our axon, essentially. We're going to go through all these specific parts that you see, but essentially it's transmitting the information to that bottom looking like monster thing that's accepting, and that's the postsynaptic cell. These can be used interchangeably. So in this specific circumstance, we would call that top monster a presynaptic neuron, but it was a postsynaptic neuron at one point when it received information from one neuron. So we talk about it in, you know, a case-by-case basis. So in this case, this top monster would be the presynaptic neuron and the bottom would be the postsynaptic. But eventually, if this creates action potentials, the postsynaptic, and sends information, then we would call it the presynaptic neuron. And neurotransmitters, which you've probably heard, you've all heard it, psychology professors, but you hear it a lot on like commercials because a lot of drugs work on the action of the neurotransmitters, but they're essentially the body's chemical messengers. So they're sent from presynaptic to postsynaptic, okay? So synapses are usually between axon terminals, so the bottom of the axons, and dendritic spines. So talking end of the presynaptic to listening end. So, all right, I don't know how much of this you can see, but like if this is one cell, this is my axon here. I'm trying to manipulate my arms in a way that they don't go. So it would essentially be presynaptic axon sending information to postsynaptic dendrites here. Sometimes we have axon to axon signals. So it'd be like this. You're like, who is this girl? Sometimes it's dendrite to dendrite, and sometimes it's axon to cell body, but we're going to focus on traditional axon to dendrite. Okay, so I typically like to start with like more basic information just to give you like the flow of what I'm thinking and what happens, and then I go into specifics. So this is more of like the elementary basic information, broad overview of what happens in communication between neurons, okay? So what happens is this presynaptic neuron, so synthesizes and packages a neurotransmitter. It could be one of a bunch of different types of neurotransmitters. We'll go into neurotransmitters in a bit. In these synaptic vesicles that are housed in the axon terminal, which is at the end of the axon. The arrival of an action potential, so going back to what we just learned, communication within neurons, causes, through a particular action, it has to do with calcium, it causes the release of the neurotransmitter. Neurotransmitter will diffuse from presynaptic to postsynaptic and is received by postsynaptic. And then, as quickly as it's in, the neurotransmitter has to be removed. And there are multiple ways that this happens. And if you think to drugs that we've heard of for certain conditions like major depressive disorder, selective serotonin reuptake inhibitors, that drug works on the action of this. We'll talk more about that, but essentially, serotonin is a neurotransmitter. Serotonin is a chemical messenger that goes from a presynaptic to a postsynaptic, and then it has to be taken away. And the name of this drug gives the information to us. It inhibits reuptake, so it keeps that neuron or neurotransmitter in the synaptic cleft longer, so the action is taking place of serotonin. Okay, so let's get a little bit more specific. Okay, so I said we have a chemical synapse and an electrical synapse. So if you look at the left, the green, you will see a chemical synapse. So you have like that space there where information is being sent across that space, okay? Give me 1 second, sorry guys. Okay, so a chemical synapse involves the release of chemical messengers known as neurotransmitters. Neurotransmitters carry information from the presynaptic sending cell to the postsynaptic receiving cell through that synaptic space, okay? Electrical is a little bit different. It's faster because there isn't that space that it has to go through. So there's a direct physical connection between the presynaptic and the postsynaptic neuron. The connection takes place in the form of a channel that's called a gap junction. This allows current or ions to flow directly from one cell to another. So we're going to go over how this can be helpful and why we have the different types of synapses. Okay, so first let's talk a little bit about a chemical synapse. So the reason I did this review is because we're going back to this information here. It's really important for students to understand the action that takes place within the neuron for them to understand what's taking place between the neuron, because it's all obviously very interconnected, okay? And these terms that we're using are they're coming up again. They're coming. These depolarizing, hyperpolarizing is coming up again right now, okay? So an action potential is going to reach the axon terminals to the end, depolarizes the membrane, opens voltage-gated sodium channels. The sodium channels enter the cell, depolarizes the presynaptic membrane. What this leads to is, interestingly, we're bringing calcium into play here. So calcium will help with the action of release of the neurotransmitter. So these voltage-gated calcium channels are open. Calcium ions enter the cell, initiating signaling cascade to cause these synaptic vesicles that are on the presynaptic neuron. They house the neurotransmitters. It causes those molecules to fuse with the presynaptic membrane, okay? So the fusion causes the neurotransmitter to be released into the synaptic cleft. So that space, so it's the extracellular space between the presynaptic and postsynaptic. So it has to take the neurotransmitter has to take a little bit of a journey across that space. The neurotransmitter diffuses across the synaptic cleft and binds to receptor proteins on the postsynaptic neuron. The binding now causes one of two actions. And when we talk about neurotransmitters, we're going to talk about this. So just hold tight here. But essentially the binding causes these specific ion channels. So letting the ions in and out. It can be an inotropic receptor, which we're going to talk about, or a metabotropic receptor. We'll talk about that. But it causes them to open. Okay. This action is what leads to if an action potential will fire on the next cell, the postsynaptic neuron, or not. Okay. So how is that decision essentially made? Well, it depends on the action that is coming in. So when we talked about communication between neurons, what I had said was that it depends if there's enough stimulus. So enough stimulus to get to negative 55 millivolts, or whatever that threshold of excitation is for that specific cell. This is a step we're at. So see how we're kind of like taking this further. So we're taking this from presynaptic, now we're in postsynaptic, and this is happening, okay? So it's going to depend on this action that takes place from the neurotransmitter if the next cell can fire, okay? Neurotransmitters all have different effects, and we're going to talk about some of the differences in different neurotransmitters. Some have what's called an excitatory effect that are more likely to lead to an action potential, and some inhibit the action of the next cell. Okay, so what we have is excitatory postsynaptic potentials, or what's known as EPSP. So that's the depolarization of the presynaptic membrane that makes the postsynaptic membrane more likely to fire the action potential. So if the depolarization of the presynaptic, so if it's making the cell more positive, that leads to more likely a greater chance that the postsynaptic will fire an action potential. But then we have inhibitory postsynaptic potentials. That's a hyperpolarization. So remember, that's going the other way. That's the cell becoming more negative. So hyperpolarization of the presynaptic neuron makes the postsynaptic neuron less likely to fire an action potential. So let's take an example of a neurotransmitter you may have heard of before, acetylcholine. So acetylcholine is really important for muscle movement. So if acetylcholine is released to the synapse between a nerve and a muscle, this is known as a neuromuscular junction, by a presynaptic neuron, so by that initial neuron, it causes postsynaptic sodium channels to open. The sodium enters the postsynaptic cell and causes postsynaptic membrane to depolarize. When that depolarizing happens, if it reaches the threshold, it can lead to an action potential. This depolarization that happens there, that's called an EPSP, and it makes the postsynaptic neuron more likely to fire an action potential. On the other hand, if you've heard of GABA, so gamma aminobutyric acid, which is benzodiazepines work on the GABA system, it leads to an inhibitory effect. So if there's a release of an inhibitory neuron or neurotransmitter, can lead to an IPSP. So it's a hyperpolarization of the presynaptic neuron. So for example, when the neurotransmitter GABA is released from the presynaptic neuron, it binds to chloride channels. Chloride's negative. Chloride ions enter the cell, hyperpolarize the membrane, less likely for the action potential to fire. So it's all this cumulative action that's working together throughout your brain. Okay, so how do these EPSPs and IPSPs interact? Okay, basically a postsynaptic neuron is going to add together everything that they're getting. They're getting EPSPs, IPSPs, and then decide whether or not they're firing an action potential. So there's two different ways that this happens. There's spatial summation, which is the integration of postsynaptic potentials that occur at different locations on the cell, but about the same time. And that can influence if an action potential takes place or not. Temporal summation is the integration of postsynaptic potentials that occur in the same place, but slightly different times. Still can influence the action, okay? So let's suppose that an excitatory synapse are made up of different dendrites on the postsynaptic neuron, okay? Neither synapse can produce an EPSP quite large enough for an action potential to fire. But if multiple EPSPs happen at the same time, they could sum or add up to bring the membrane potential to the threshold of excitation. On the other hand, if there's an IPSP that occurs together with one or two EPSPs, it might prevent the membrane potential from reaching a threshold and keep the neuron from firing an action potential. Okay, these are examples of spatial summation. So occurring basically at the same time. So in terms of temporal summation, a key point is that postsynaptic potentials aren't instantaneous. Instead, they last a while before they dissipate. So you could have a presynaptic neuron firing quickly twice in a row. I mean, remember, it has to go back to baseline. Essentially, it has to go back to rest before it can fire again. But you can still have them quickly fire one after another. And if one EPSP, say it arrives on the postsynaptic, takes a little while to dissipate, before it dissipates, another EPSP comes in. Could help bumping the membrane potential above threshold, okay? So it's essentially, these are both cumulative effects, but it's EPSPs, IPSPs occurring at the same time versus occurring at slightly different times, but still leading to a specific action on the postsynaptic neuron, or no action. If, say, there's an EPSP hanging out in an IPSP, it's like, yeah, it's not gonna happen. Yep, I just did that. All right, so I think we've all been in our house for a little bit too long, guys. What do you think? All right, so I said that the neurotransmitter has to be removed from the cleft. It can't hang out there forever. There has to be a removal system so that the action can keep taking place with new cells. So there are three ways that this can take place. So let's go back a little bit. So after a neurotransmitter has been recognized by the postsynaptic cell, it's released back in the cleft. Like, that was quick, thanks for doing the job. Once it's in the synapse, it's quickly removed or chemically inactivated to prevent constant stimulation. So say that you have an excitatory neurotransmitter, and it's just continuously creating EPSPs and continuously creating action on the postsynaptic cell. Sounds like things could go away. It needs to do its job, and then off it goes. kind of like me today. You don't want to hear me all day. It would probably be really exhausting to listen to me talk for 8 hours, and I would be very hungry because I wouldn't eat. I would just be talking. So just do my job and then go away, right? So this is what happens there. This is what happens here. So a neurotransmitter may be broken down by an enzyme. It may be sucked back up into the presynaptic neuron, or it could just simply diffuse away. So diffusion, neurotransmitter will diffuse from the synapse into a tissue fluid away from the synapse. It gets absorbed, actually by glial cells, which are the other main nerve cells in our body, or they're the other main cells in our body besides nerve cells. Degradation. So an enzyme can break down that specific neurotransmitter, making it inactive. And then those breakdown products are recycled to be reused. So think about acetylcholine again. There's an enzyme called acetylcholine esterase within the synaptic gap, and that can help break it down and recycle it to be used again. Okay. And then there's the recycled reuptake. So this is where we're going back to when we hear about SSRIs, selective serotonin reuptake inhibitors. This is what it's working on. I always thought this was cool. I bring up this example a lot, but it made me, when I was first learning this, it made me connect. Like I was able to connect because that selective serotonin reuptake inhibitors, there's also selective norepinephrine reuptake inhibitors. The way that the name was structured, I was like, okay, this is telling me exactly what's happening and where it's happening. So I understand that it's taking place, the action of the neurotransmitter, and it's basically helping more the cell to fire or to not fire. It's really, creating that chain of events. I just thought that connection was really cool. So transporters are proteins that are embedded in the presynaptic membrane. Okay, they're able to pump neurotransmitter molecules from the synaptic cleft back into the presynaptic cells. So this is the process that's called reuptake. It's typically classical to all small molecule neurotransmitters. We're going to talk about that, but some you may think of off the top of your head. Like if I tell you to name neurotransmitters, it may be this list. Like there are the common ones you think of: glutamate, GABA, dopamine, our pleasure hormone or our pleasure neurotransmitter, epinephrine, norepinephrine, serotonin. Okay. So we're not going to spend too much time on core electrical synapses, but I want to just briefly talk about it. So in an electrical synapse, the presynaptic and postsynaptic membranes are physically connected by channel proteins forming what's called gap junctions. Okay. Gap junctions allow the current to pass directly from one cell to another. So there's not that like space and that whole like process that can take place within that space. And the signaling that takes place in electrical synapses is virtually instantaneous, as opposed to chemical synapses that takes a little bit longer. They're more reliable because they're less likely to be blocked by anything, and they're really important for synchronizing electrical activity of a group of neurons. So for example, electrical synapses in the area of the thalamus, which is the main relay station of the brain, are responsible for a ton of different things. But one is sleep-wake, and seizures can be associated with those. So these are thought to regulate slow-wave sleep, so helping us to ensure that we're getting adequate sleep, and any disruption of these can lead to seizures. So what are the benefits? So for one thing, they're fast, which can be important. So say there's a circuit, our brain, we need to escape a predator, that fight or flight response. This can create a really fast circuit that allows us to fight or flight or flee. And it allows for synchronized activity for a group of cells. In many cases, they carry electrical current in both directions. So the depolarization of a postsynaptic neuron will lead to depolarization of a presynaptic neuron. So this a little bit bends the definition of a presynaptic and postsynaptic neuron, but essentially it can happen both ways in this case. The downsides are, unlike chemical synapses, electrical synapses cannot turn an excitatory signal in one neuron into an inhibitory signal in another. So they lack some like versatility, flexibility, and capacity for signal modulation that we see in chemical synapses. So chemical synapses essentially like kind of slowing it down a little bit. and like allowing for more like precision and more like the accuracy of the signals being sent. Okay, so we have about 5 minutes left or so, and we're going to talk a little bit about neurotransmitters and receptors. So receptors are located on the postsynaptic cell, and that's what's essentially like catching the neurotransmitters. There's different types, just because we want to make it more complicated, right? You can't just make it simple here. So we have what's called ionotropic receptors and metabotropic receptors. So these are on the postsynaptic cell, and these are essentially like what's capturing the neurotransmitter that's being sent from the presynaptic to the postsynaptic. So ionotropic receptors are membrane-spanning ion channel proteins that open directly in response to some sort of binding. They act to quickly depolarize the neuron and pass on the action potential, or hyperpolarize, okay? So they're kind of fast acting. So the neurotransmitter work on the presynaptic, the ionotropic receptor doesn't need any help, essentially. So a neurotransmitter can be sent straight to the postsynaptic cell and act on it. Metabotropic need a little bit of help. So they themselves are not ion channels. The neurotransmitter binding triggers the signaling pathway that may indirectly open or close channels and or have some other effect entirely, but they need the help of some other actions that take place. So they're called metabotropic receptors because the eventual movement of ions depends on one or more metabolic steps. So the receptor complex itself includes this receptor protein that we call G protein. The neurotransmitter, referred to as the first messenger, binds to the receptor protein at the extracellular surface of the cell, and the intracellular side of the protein initiates the action of the G protein. So the G protein kind of helps that signal being sent. So the receptor itself needs a little bit of aid, so it's a little bit slower action that takes place. So ionotropic receptors undergo a change in shape when the neurotransmitter binds. That causes the channel to open and the neurotransmitter flow through, and then you get depolarization or hyperpolarization, depending on the type of neurotransmitter. It typically produces a very quick physiological response, and then the neurotransmitter is able to be removed from the synapse very rapidly, which in most cases, it gets a little bit complex depending on the type of neuron and the action that's taking place, but you want it to be taken back up so that the system can keep functioning as it is. So the flow starts within tens of microseconds with an ionotropic receptor, the neurotransmitter is binding. All right, so metabotropic receptors, the protein to which the neurotransmitter binds, not the neurotransmitter receptor, is not an ion channel, okay? So this receptor depends on the activation of several molecules inside the cell and often involves what's called the second messenger pathway. So that's what I was saying is like kind of the helper system there. So the ion channel can't open on its own. It needs a little bit of help there. It's a slower signaling than ionotropic receptors. And it seems, that seems to make sense because it needs a little bit of help. So now let's get into the main event. All right, so neurotransmitters. So neurotransmitters are essentially the body's chemical messengers. So this is when we're talking about chemical synapses. They're molecules used by the neurotransmitter system to transmit messages between neurons. The signaling at a synapse brings about the response that depends on both the neurotransmitter from the presynaptic cell and the receptor at the postsynaptic cell, which we just discussed. A single neurotransmitter may have more than a dozen receptors. So what we know, there are so many different types of neurotransmitters. I'm not going to go into all of them. New ones are still being discovered. I'm just going to kind of go over like the ones that are more common. So keep that in mind that I'm not going over everything. You may go more in depth with your students depending on what the class is. And you may go in more in depth into the specific actions of the neurotransmitters. I'm not really going to go into that. I'm going to go more so into what they're used for. So let's go over a few common ones. So conventional neurotransmitters, so dopamine, GABA, serotonin, the ones we think of are the conventional neurotransmitters. They share certain basic features with one another. They're stored in the synaptic vesicles of the presynaptic neuron. They get released when calcium enters the axon terminal in response to an action potential. So from the help of calcium after the action potential, that's when they get released. And they act by binding to receptors on the membrane of the postsynaptic cell, okay? So that kind of goes along with everything that we've been talking about, not too complex, right? So a lot of the common neurotransmitters are called small molecule neurotransmitters. Here are the three main groups of those. We'll briefly go into each. There's acetylcholine, which is kind of a loner, amino acids, and biogenic amines. So acetylcholine is a common neurotransmitter in both invertebrates and vertebrates. It's vital for functions involving muscle stimulation, which we talked about. If there's an impairment in the acetylcholine system, there could be impairments in muscle stimulation. Memory formation is really important for learning. We, as vertebrates, have two major classes of acetylcholine receptors. One is ligand binding, or another word for that is ionotropic receptor, and one is metabotropic. Amino acids, glutamate and GABA, what we are common, they're not all of them, but they're common amino acids. So glutamate is the most common brain neurotransmitter. It regulates the development and creation of new nerve pathways, it's really important, and hence it's involved in learning and memory. So these new pathways are created in response to things that you're learning. So today, if you're learning, this could be a review for you, if you're learning something new, glutamate's helping you out there, okay? It's an excitatory neuron. GABA is an inhibitory neuron. It's the most abundant inhibitory neurotransmitter in the brain. So it inhibits firing of the central nervous system. High levels can improve focus. So having that in that space, that synaptic space, can be a good thing for inhibiting the action of the other cell. Low levels can lead to anxiety. A little bit of a simplistic explanation, because we think of the action of benzodiazepines, which work on the GABA system. They're typically prescribed for sedative properties and anxiety. So lorazepam. They do not act directly on the receptors, but they bind allosterically to GABA receptors. The results are an increased probability of a channel opening and potentiation of GABA neurotransmitters in the brain. So it gets a little bit more complex, which is what I was saying, a little bit too much for this hour-long talk, but if you go into this with your students, there are a lot more specifics when it comes to the different types of neurotransmitters. Biogenic amines are active in both the central nervous system and the peripheral nervous system. They play a role in several nervous system disorders and treatment. You often think of dopamine being associated with Parkinson's disease, with schizophrenia. Depending on the action, can be associated with one or the other. So just some examples: norepinephrine, predominantly a neurotransmitter that's occasionally released as a hormone. It contracts blood vessels, increases blood flow, and it can help improve attention and speed at which a response occurs. Epinephrine is primarily a hormone that's released by the adrenal gland, so in the peripheral nervous system. Some neurons do secrete it as a neurotransmitter. Helps increase heart rate, blood flow, leading to a physical boost and heightened awareness. So epinephrine, adrenaline, synonyms. So you think of that, the fight or flight response, the sympathetic nervous system, the release of epinephrine kind of like gets you ready to take action. It's produced during stressful or exciting situations. And then we have dopamine, which is primarily responsible for feelings of pleasure. It also is important for movement and motivation. People tend to repeat behaviors that lead to dopamine release. So the other day I made chocolate chip cookies for lunch because quarantine is weird, and I ate 3 of them. I had 3 of them because every time I ate one, I had a dopamine release. So the action of dopamine, I just wanted to keep eating cookies. I'm sure many of you can relate because chocolate chip cookies are amazing. Unfortunately, if this goes awry, it can lead to addiction because of the action of dopamine. We are seeking that pleasure that we get from the action of dopamine. Serotonin, we know that it's associated with depression, but we know based on the action of medication that inhibiting immediate reuptake can help with depression, but we don't know if serotonin is the cause of depression. We just know there's an association there, but it can contribute to feelings of well-being and happiness. It's affected by exercise and light exposure, so both important things for us as I'm sitting in a dark room, not exercising. And it also can play a role in the sleep-wake and digestive system response. So we're going to end on those. Can be problematic, but for the most part, heavy neurotransmitters. And I encourage you to eat chocolate chip cookies for lunch and then exercise. So that's all I have. Thank you so much for having me. I really enjoy speaking for Hawks. I hope that this information is helpful to you and not review, or, you know, it's probably a review for some of you, but I do hope that you walk away, hopefully at least learning something. If you have any questions, feel free to let me know. I also posted, I try to keep, you know, my university job a little bit separate. So if you do have questions, please feel free, send me an email. I'm also always happy to talk about online teaching. I had a transition. I didn't have to, from a traditional classroom to online teaching. It took me a lot to learn, and Danny knows that I don't stop talking, but when I first joined the university, then I started serving on the teaching and learning committee, which helps with professional development for teachers, and I've learned so much, and I really, really love teaching and learning. So if you ever have questions about online teaching, I love meeting with people, especially now when we're all missing social interaction and chatting. So I'm always happy to talk. So please do feel free to get in touch with me. So that's all I have. Thank you so much.
All right, thank you, Dr. Slifka. That was great. We are getting some comments in the question and answer box already. We have a few instructors just thanking you for the content of the presentation.
Of course, thank you for coming.
So we also had Maria ask if there will be a recording, and the answer to that is yes. We will be sending out a recording. It'll be posted on our blog. Also on the blog are the other 2 webinars that Dr. Slifka has done for us. So let me try to look through. We are getting a cascade of just thank you, great presentation. This was so helpful. I didn't know anything about electrical synapses, so thank you. More thank yous. Just trying to work my way through these.
Yeah, I'm trying to keep up with them too. I really appreciate all of this. It's so nice. Just like, it's a different audience than what I'm used to. I'm used to, you know, my students, and I love my students, but it's great talking to colleagues as well. I'm glad that it's helpful. I want this to be helpful. I don't want you guys sitting here and just listening to me blab for an hour and then walking away and saying, hey, none of that really helps me enough. So,
and Dr. Slifka, just so you know, I'm sort of filtering some of these into the dismissed column. I think you can probably see that, just to make sure we can address any questions if there are. I'd love to hear it, just if you have any short tips for some of the online teaching, just anything that you've learned with this transition that maybe somebody can take away as they're starting to think about moving their courses online for summer or even for next fall as this situation stretches on.
Yeah, I think it all depends on the platform that you're using, but I think what I've seen with my students, and I try to be funny during these, but I'm not the best. I mean, you know, you try to do your best, but I think students can sometimes zone out, you know, especially if they're at home and they're in their pajamas and they're watching this lecture. It's very easy to like just kind of like do other things and zone out. So I think the incorporation of things that you would do in a traditional classroom. I guess online is now a traditional classroom, but some things that I do in mine are role play. So say if you're teaching the history of psychology, everybody adopts a different character, or not a character, a different person. And they communicate as if they are that historical figure. You can do debates. You can do live polling. So incorporating things that allow students to have more of a similar interaction they would in a traditional classroom. So it makes the transition a little bit more seamless and they're still learning. That's what I found to be beneficial. Oh, Danny, I think you might be on mute.
I am, yes.
Okay.
I think we have time for one more question. We have a content question here that asks, is the medial forebrain bundle an example of axon to axon communication?
Oh, am I doing that background stuff? Sorry.
Would inhibiting one pathway inhibit all pathways given their close proximity?
So that's a good question about the medial forebrain bundle. And to be honest, that is going way back to my neuroscience training. And I'm completely honest. I'm completely honest with my students. I don't remember the answer to that one. I'm happy to discuss. We can look it up together. In terms of the inhibition, no. So it depends on the action of each individual neuron. So it doesn't mean that an entire pathway would be inhibitory. It could mean like components of the pathway and that specific signal could be inhibitory. That helps.
Okay, thank you.
Yeah.
Well, thank you again to our audience for joining us today. Thank you, Dr. Slifka, for the presentation one more time. If you have questions for us at Hawks Learning, you can email us at marketing@hawkslearning.com. And I will be emailing everyone later on today or tomorrow with the recording once that is processed and hosted on our website. So hope everyone has a great rest of their day and stay safe.
All right. Thanks, Danny. Thanks, everybody. Have a great week.